most of you have seen some of the material on this particular study. This is a coupled study really looking at vegetation dynamics and hydrologic responses to mountain pine beetle attack. And really we did have last year about this time, a half day, actually it was a full day session with the TRIA program sort of reporting on the data. So today really is just going to be kind of a high level overview of some of the results that the two researchers that principally drove the lion's share of this work, Anne McIntosh and Pablo Pina, who's got a real job down in Calgary these days, did. So really quickly, I'm not going to spend much time here. From an ecological and a hydroecological standpoint, the reason we're interested in mountain pine beetle from a disturbance perspective is it's really a novel kind of a disturbance agent. You know, we have overstories that are killed, but really without the understory disturbance in place. And that sets it apart from how we understand ecological processes in harvested systems or by wildfire. And, you know, that sets up some really interesting questions. Well, what happens after mountain pine beetle attack? How does the floristics, the vegetation in the site, respond in the short term? Because that's going to set up some of the initial trajectory for how the vegetation responds in the longer term. And this has really got some tight relationships with how hydrology is sort of reflected. Alan, you alluded to the fact that there's some uncertainty in terms of hydrologic responses. And that comes back to, I guess, some of Barry's points. It's in the details. Some of my colleagues from British Columbia have spent a lot of time trying to model these hydrologic responses using uncertain kind of relationships between precipitation regimes and how hydrology in these varied landscapes works. Some of my colleagues in BC Ministry of Forest and Range have also tried to do some of the work on the ground and have not been that successful for a number of reasons, and I'll allude to those in a second. So really, basically, this program had two major sort of components or pieces to it. So one was really looking at how is the hydrology of these affected stands impacted, and then the companion parallel piece is, you know, looking at the early trajectory of vegetation responses, because that's going to sort of set up where things go in the future. My colleagues in BC Ministry of Forest and Range certainly tried to tackle a lot of these problems. And part of the difficulty they had was, as the beetle attack was really getting underway, they were studying the impact on vertical water balances in these affected stands, but at the same time they were losing their reference stands. And so that really limited their ability to provide these insights. And so that really was our ability to kind of learn from the challenges our colleagues had faced before. So we really approached this from a very different sort of approach, is really trying to simulate mountain pine beetle attack using glyphosate, single tree injection of glyphosate, to reproduce the gross canopy scale effects of tree death, followed by sort of crown reddening, and then presumably moving into latter phases of the attack. Because really it's the death of that crown that drives the changes in light regime, that drives the changes in the hydrology. So really we tried to sort of look at a number of things. We had some control stands. We treated some stands where we tried to treat about 50% of the trees in a distributed fashion. And then other stands where we treated all of the trees, trying to produce a more severe attack. And then another treatment where we were looking at some harvesting stands, really trying to get some comparative stuff on what would happen after these stands are harvested. This is the basic work was set up here south of Rob. Series of plots. These are large plots, 2.2 hectare plots for the hydrology component of this. So we treated a lot. We killed a lot of trees. And then some additional plot pairs, replicate groups, down further to the south. And basically the approach was a classic before-after control impact study design. So a classic experimental design where we're looking at both treated trees and treated plots, control plots, both before and after. So we can really disentangle with a fair bit of resolution. And the work took about 4 years. 1 year first installing the equipment, and then sort of 1 year of pre-treatment measurements, and then a couple of years of post-treatment measurements, really to disentangle what was going on. At the end of the pre-treatment year, basically the glyphosate treatments were applied, and as I said, we killed about 8,000 trees, and the harvest treatments were applied at that time, and then 2 years of post-treatment measurements. And just a quick snapshot here. The glyphosate worked pretty well. It was applied in midsummer of 2009, and even by the end of that season, we were starting to see crown reddening. The next year, we started to see sort of both the chlorotic signs of sort of the late phase green attack, but also starting to see crown reddening. And here, just to give you an idea, here we have control, 50% kill treatment, 100% kill treatment. The green are the control sites, and the orange bars are the treated sites. So here's the control. Here is the 50% treated site and the 100% treated site, and we're looking at healthy basal area. So we got pretty close to the sort of target treatments, not exact, but during the course of the study, we still had those dead needles retained. So really, this study over a 3.5 year sort of measurement period really is representative of that earlier phase, green attack through red attack phase. So in interpreting the work, it's really characteristic of those early green attack, red attack sort of phases. Really quickly, I'm not going to spend much time here. The hydrology work was basically, we think about some of the things that Alan alluded to. The hydrologic effects that people are worried about, how is stream flow affected, timing of runoff, flooding issues, a number of those kinds of things, they're all driven by how the death of trees after mountain pine beetle attack affects cycling of water sort of at the stand scale. And so that really became the focus of the hydrologic work. What is the rainfall interception? This is the amount of the precipitation that's caught by the foliar surfaces, branches, re-evaporated back into the atmosphere that never reaches the ground level. And then what is the interception that's taking place in the forest floor, understory layers? Those are really evaporative losses along with the transpiration, because at the end of the day, you've got inputs of precip and you've got evaporative outputs. What's left over is stored in the soil or contributes to groundwater or soil recharge. And so that's really the measurements that we're focused on. Very quickly walking through this. On the rainfall interception side, we found that unlike previous work, rainfall interception in these mature stands was actually a fair bit higher than we thought. The canopy was using about 40% of the total growing season precipitation. The forest floor intercepted about another 30. So we're looking at about 70% of the growing season precipitation that's basically lost back to the atmosphere. The storage capacities for these pieces are quite high, so you can interpret this as you actually need some pretty big rainfall events to overwhelm those capacities and recharge the system. And to cut to the chase, because of needle retention during this green and red attack phase, we found during that early phase there really was no impact of the treatments on rainfall interception. Transpiration was a different beast, though. We did spend a lot of time arguing and throwing chalk brushes at one another, trying to figure out, well, what actually is tree death? What's the physiologically evidentiary basis of tree death as it related to transpiration? So we did actually do some measurements on dead trees, and in fact, the dead trees didn't transpire, so no big surprise there. The fading chlorotic trees, of course, we did see reductions in transpiration. When we sort of looked at that at a rate basis, it's about 1/3 reduction in transpiration during the early fading pieces. The interesting thing we did see is the trees, the healthy trees that were surrounded by fading or dead trees, because there was a soil moisture signature, those trees had more moisture resources available to them, and their transpiration rates actually increased by about 35%. And so when we scaled that up at the stand scale, really it's the composition of dead, fading trees and live trees within the stand that really governs the overall vapor fluxes. And so in this 50% mountain pine beetle treatment, we found that those stands were actually using about 10% more water than a comparable healthy stand because of this sort of, you can almost think of it as a thinning effect, but it really isn't a thinning effect because the canopy hasn't been altered. whereas the mountain pine beetle, the 100% mountain pine beetle affected stands decreased their water use by about 50%. Soil moisture, really for these, we saw that most of the soil moisture signature was kind of constrained to the surface. First 0 to 5 centimeter layer or 0 to 20 centimeter layer was really the strongest signature. And we really didn't see much sort of when we started to consider deeper horizons. So sort of a surface focus to that. Basically, all of these pieces of information were wired into kind of an integrated hydrologic model that's looking at transpiration and interception, using kind of the climatic variation along the Lodgepole pine-dominated region of the province to drive it. And I guess the one main thing was, because these processes interact in non-linear ways, the responses aren't really easy to sort of predict. There was a number of different scenarios that were looked at. One sort of partial attacks, more massive attacks, and then gray attack in terms of sort of modeling. And this here really is just looking at the overall consequences in terms of groundwater recharge that would really drive hydrologic change. And so here you can see these partial attacks produced smaller increases than the gray attacks. This is sort of in millimeters per season, but these are the proportional changes from undisturbed conditions. So you're seeing fairly large changes in generated runoff, 50% to 250% to over 500% increases in groundwater recharge. And in terms of this piece across the province, climatic variability, we saw that there was a greater effect where growing season precipitation was greater. I'll turn this over to Ellen.
Okay, so I'm going to talk about the post-attack vegetation and below ground responses, and then spend more time looking forward. So I can't remember if Uldis mentioned, but there are two posters here, one by Pablo and one by Anne, and so all the details of the research results can be found there. So the part that Anne did, she should be giving this because it was her thesis, but she was looking at what the early trajectories are of post-attack vegetation and the below-ground responses after these different treatments and in the early red attack phase. So this included looking at forest structure, understory plant communities, pine regeneration, and below-ground aspects, including microbial communities and soil nutrients. So this is what we anticipated would happen when we looked at this from the beginning. First of all, we knew that the trees remained standing, and so the understory, and they retained their needles. So the understory microclimate was not going to change much in the short term, so that wouldn't be having an influence on the understory. There's no direct disturbance of the forest floor, so the feeling was that probably there aren't going to be new microsites for pine regeneration established, and the understory vegetation itself was basically left untouched, and so it was only going to respond to whatever possible above ground, below ground resource changes it would be experiencing. We knew that tree transpiration would be reduced, so we thought there'd be increased soil moisture and nutrient availability, and this might feed into a response of the understory vegetation. There probably wouldn't be new inputs into nutrient cycling because the litter inputs would be delayed until the foliage started coming off the trees. So that was kind of the outlook from where we started. And of course, as we've said before, that this was a very different type of disturbance than we get with wildfire or with harvesting. So we expected the salvage logged treatments to behave much differently than the MPB treatments. Okay, so quickly the methods characterizing the forest structure. This included the tree species, whether they were live or dead, the canopy cover, looking at the down wood accumulation, looking at the understory plant communities. So this is all the wonderful plants that live underneath the trees. Shrubs, forbs, graminoids, bryophytes, lichens, which aren't plants. Quantifying the below ground aspects, the soil microbial communities and soil nutrients, and then looking at seedling samplings, the extent of advanced regeneration, and more particularly, and did a sowing experiment to look at what the future possibilities, or what the possibilities would be for natural regeneration of pine in these sites. And as I'll just mention, we had a pre-treatment year in 2008, and then up to 2010, or in some cases looking back up to 2012 in terms of collecting data after the treatments. Okay, so very quickly the results with no graphs. The graphs are all on the poster. In terms of forest structure, the story was, yeah, the trees were dying because we injected them with herbicide, and it was more in the mountain pine beetle plots than in the other ones, and this resulted in a reduction in live basal area, and Uldis already showed those results, but the canopy cover remained unchanged. So because the needles were retained to the hemispherical photos, which you can see here, the hemispherical photos there that were used to quantify canopy cover, so there was really no change in canopy cover during this time period. And downwood was slowly accumulating, so the treatments were kind of on top of a process of death and senescence already occurring in these mature lodgepole pine stands. So the trees were dying anyways, even if we hadn't treated them, and downwood was accumulating, but it was relatively slow because the treated trees remained standing for the most part. In terms of the understory plant community, there were minor changes in species richness and in cover. It increased slightly in the mountain pine beetle treated and declined in the salvage harvested treatments, but these were pretty minor and mostly not statistically significant. And the composition changed quite dramatically in the salvage harvested with the increases in the shade intolerant and early successional kind of species, and that was not unexpected. But the mountain pine beetle treated stands really didn't change their composition much. In terms of the forest floor microbial communities and the soil nutrients, there were fairly dramatic changes among years, but these were independent of any treatment effects. So we didn't see the below ground changes that we expected to accompany the increases in soil moisture that we'd seen. So we did see the increases in soil moisture, but this didn't translate into increased nutrient availability in the stands, and it didn't translate much into changes in the soil microbial community, so no real signal in terms of changes in nutrient cycling or below ground resources. In terms of regeneration, the stands that we picked had a low amount of advanced regeneration in them, and the natural occurrence of regeneration was very poor. Even when we did the sowing experiment in different microsites, the survival of those seedlings and germination to survival over a few years was quite poor. In general, the survival of the sown seeds, or germination survival of the sown seeds, was better in the salvage harvested treatment than the NPB treatments, and it was better on the mineral soil and decayed wood. So those were the microsites that were best for natural regeneration of pine, but unfortunately these are microsites that are not very available in these sites. So the dominant microsites, the microsites with the thick moss and heavy herbaceous cover were not good at all for natural regeneration of lodgepole pine. Okay, recap and looking at the future, where we're going with this. So just a reminder of the timeline. We were doing setup in 2007-8, and then in 2008 up until June of 2009, pre-treatment data collection, applied the treatments in the last 2 weeks of June in 2009, and then followed for 2 full years post-treatment. And last summer and this coming year, we're in a monitoring stage, so with a small amount of funding to continue some baseline monitoring at these sites. And then we're hoping to receive funding to go into phase 2, starting with full sampling again in 2014. Maybe like if we had some money to do a little bit more work this coming summer, because it's been quite a gap since we had a full assessment of some of the variables. But we want to look ahead now and follow this into the gray attack stage. Fortunately, the sites haven't been hit with mountain pine beetle, so we still have our control and we still have our gradient of mortality caused by the treatments that we applied. So what are we expecting in this next gray attack stage? So first of all, I'll just explain that interception was dominating the stand water balance, so 70% of the growing season precipitation was lost to interception, 40% in the canopy and 30% in the forest floor. Therefore, we expect that once the needles start to fall off, we're going to see the major hydrologic responses once that interception is declining. So when the needles are shed, we're going to see probably much more dramatic hydrologic responses and changes in the stand water balance, and this is not something that's been documented previously. And so we want to ask how rainfall interception dynamics change as we move from the red attack to the gray attack phases, and then canopy interception, what exactly happens to canopy interception. The trees are still standing, but as they begin to lose their foliage and branches start to fall down, we anticipate canopy interception will go down a lot. And then what will happen in the forest floor interception, and that feeds into how the vegetation, the understory vegetation, might respond to other changes that are happening in the stand at the same time. So they may intercept the same amount, they may start to intercept more of the precipitation. We don't really know what their capacity is to compensate for the loss of interception and transpiration in the canopy. And then, of course, linking these hydrologic changes to the soil moisture responses and the ability for the soil to store moisture, what the drainage will be, and what the groundwater implications might be of those changes in stand level water balance. So the impact of this work will be that, combined with the work that Uldis already explained, it'll provide insights into the full picture of the sequence of hydrologic changes as we move from green attack to red attack to gray attack stages. In terms of vegetation in the below ground, we know as the interception declines, soil moisture and nutrients should be increasing. So below ground resources will be increasing, and then as the foliage is shed, the understory will become warmer with more light. So we're expecting to see more dramatic responses of the understory to this release of above ground and below ground resources. And we don't know exactly how they'll respond. I mean, probably they should be increasing in cover, potentially in richness. We'll have more shade-intolerant species or increased abundance of shade-intolerant species. And we don't know how that will play out in terms of how that might compensate for the interception and transpiration that's lost at the canopy. So the understory may intercept more of the vegetation, sort of ameliorating the hydrologic impact of the loss of the canopy. They may be able to increase their leaf area and transpire more, again, ameliorating the impact of the impact on the canopy, but we don't know, and we need to be able to quantify that. As the branches and the cones fall down, it's likely that a number of those cones will open as they are shed on the forest floor, and if light's getting down there, they may be warm enough to open up. And there's some evidence that the cones may open on the trees as well, on the dead trees over time. There might be some minor disturbances of the forest floor, maybe trees start to fall down and stuff, but really generally I don't think there'll be a lot of disturbance that's going to create great microsites for pine regeneration. And so it'll be very interesting to see what the potential is for natural regeneration of pine in these stands as the canopy starts to move into a gray attack and break up beyond that. Litter inputs will increase as the needles fall, and that can feed into the nutrient cycling, but maybe not very quickly, since the needle litter is going to be fairly recalcitrant. Okay, just a quick review of the deliverables of the project. We had two great field tours of the area, one for the ASRD staff, and a group from Mexico came out and saw the sites. They're kind of cool to look at if you have an opportunity to get out and see them, or if we're there, we're always happy to show people around. A number of presentations, a brown bag lunch, and then the Fry Mountain Pine Beetle Ecology Workshop in 2010, 2011, and last year as well. And then last year as well, we had the major closing workshop for the first phase of our project, jointly with the TRIA project. And then, of course, this presentation today. Quick notes, we have 3 quick notes that were produced that are available from Fry, and also our final report, which I think is available from Fry. We gave it to them, and they can give it to whoever they want to. So there's more information if you want it, you can get from these sources. And then we have publications, of course, the 2 PhD theses from Anne and from Pablo, and then 3 publications that Anne has in press or published already that are here. And if you want copies of these, you can just ask, and we'll send you illegal copies of them. And more publications are coming along with Pablo, and we're hoping to produce a synthesis publication for Forestry Chronicle. I'm not exactly sure when we're going to get that done, but we're hoping to produce an overview of the whole project and publish it in Forestry Chronicle. So that's what's there, and thanks for the support, Fry and FRIA, ASRD, West Fraser, NSER, CONACYT, scholarships for support for the students, Milo Mihailovic for helping with the herbicide treatments, many field assistants. Yeah, and if you want to contact us, there's our emails. So